Hello students, today we are going to discuss about the glycolysis and citric acid cycles. What are the steps in the glycolysis, what are the steps in the citric acid cycles, what are the steps in the glycolysis, what are the steps in the citric acid cycle, what are the enzymes involved in the glycolysis, what is the importance of the citric acid we are going to discuss. The glycolysis is very important metabolism in the body, in the every cell also. So, glycolysis is a consist of the two phases is there. The first phase is the glucose breakdown to the two molecules of the glycerol 3 phosphates and series of the 5 reactions is there. In the second phase, the another 5 reactions of these two molecules glyceraldehyde 3 phosphate molecule and final product is pyruvate. So, the glycolysis is a series of reaction extraction energy from the glucose by splitting up into the 3 carbon molecules of the pyruvate. So, the glycolysis first molecule is the glucose, that is a 6 carbon containing molecule. The end product is called as a pyruvate, is a 3 molecule containing a molecule. The glycolysis process is glucose breakdown a procedure produce the energy. It produces 2 molecule pyruvates and ATPs and NADP and water molecules is the end products. The glycolysis is a metabolism pathway converting the glucose into pyruvate, and in the most organisms occur in the liquid part of the cell of the cytosol region only. Okay. Now, next this is the glycolysis pathway metabolism. Now, this is the first molecule entered to the our cytosol region. This is the glucose molecule, it is 6 carbon containing molecule. Now, in the presence of ATP and it is will converted to the ADP molecule in the exokinase enzyme. So, it will converted as a glucose 6 phosphate molecule. Then, in the presence of isomerase enzyme, it will convert to the fructose 6 phosphate. This is the isomerization point of the phosphate molecule, and another is a fructose kinase enzyme is there, that is fructose 6 phosphate molecule to fructose 1,6 phosphate molecule conversion. The position of the glucose, the carbon 1 position is a phosphate group attached there, and 6th carbon position is carbon phosphate group is attached there, that is called a bis position. Di is different and bis is the different. Bis is the different locations, the di is the side by side molecule attached to the phosphate group, but that is why we are calling as a fructose 1,6 bis phosphate molecule. Phosphatase, this is going to fructose molecule is break down to 2 molecules of the glyceraldehyde 3 phosphate and dihydroxy acetophosphate molecules. This is the different enzymes is involved there. Then it will convert it to the glyceraldehyde 3 phosphate dehydrogen enzyme. In the presence of glyceraldehyde molecule, it will convert to as a 1,3 bis phosphate glycerate molecule. Now, you see in the 6 carbon molecules, so it converted to 4 carbon molecule and 3 carbon molecule of the dihydroxy acetate phosphate molecule. Then it will isomerization and reversible reactions is there in the presence of the enzymes. Now, the glycerol 3 phosphate molecule is going to enter to the converted to the 1,3 bis phosphate glycerate molecule. Now, again phosphoglycerate kinase enzyme is there in the presence of ATP and ADP to ATP conversion. The removal of the 1 phosphate group is attached to the ADP molecule. That ADP molecule is converted to the ATP molecule of the energy source. Then, in the presence of phosphate glyceraldehyde phosphate kinase enzyme, then it will convert to the 3 phosphoglycerate molecule. So, now it will come as a 3 carbon containing molecule, that is called a 3 phosphoglycerate molecule. Now, another enzyme in the presence of phosphoglycerate molecule, it will convert it to 2 position of the phosphate group attached there, that is called phosphoglycerate molecule. It is removal of again it will remove of the water molecule, that is called converted to PEP molecule, phosphoenol pyruvate molecule. That is, this is the also 3 carbon containing molecule. The finally ADP is attached to the in the presence of pyruvate kinase enzyme, it will convert it to the as a pyruvate molecule. So, the total reaction is coming to glucose to pyruvate molecule. In this 2 reactions is there, first reaction and second reaction is there, that is called 2x molecules. then it will convert it to the pyruvate of 3 carbon molecule. Now, the first step we are going to very deeply discussion about the first step, phosphorylation of the glucose molecule. The glucose is phosphorylated with ATP to give the glucose 6 phosphate, and isomerization of the glucose 6 phosphate to fructose 6 phosphate. The isomerization of the glucose 6 phosphate in the second reaction of the fructose 6 phosphate occur in a ring opening of the subsequent to the keto-enol taumerization process, and third step is called as the phosphorylation of the fructose 6 phosphate. The third reaction, the another phosphorylation reaction is there with the ATP, whereby the fructose 6 phosphate is converted to the fructose 1,6 phosphate. And fourth step is called fructose 1,6 bisphosphate to glyceraldehyde phosphate and dihydroxy acetone phosphate molecule. A key branch reaction in the fourth reaction to ring opening the fructose 1,6 bisphosphate, which is cleared in a retro-aldo reaction into the glyceraldehyde 3 phosphate and a dihydroxyacetone phosphate molecule. Why we are calling as a retro-aldo reaction? This is the aldehyde containing and keto conversion is there. The conversion of the ketone to aldehyde, aldehyde to ketone in the presence of enzyme. So, that is why we are called as a retro-aldo reaction. Now, fifth molecule is going to isomerization process of the dihydroxyacetone phosphate molecule. This is the isomerization mechanism to the conversion of the glyceraldehyde phosphate from dihydroxyacetone to glyceraldehyde phosphate. The branch via dihydroxyacetone phosphate is a challenged break back into the glyceraldehyde 3 phosphate into the fifth reaction of the isomerization reaction. Now, the glyceraldehyde phosphate oxidation process and phosphorylation to the 1,3 bisphosphoglycerate molecule, that is called as a sixth reaction. The combined the D-glyceraldehyde and 3 phosphate from the both routes in the oxidase at the C1 to carboxylic acid, and then phosphorylated into a one position of the yield 1,3 bisphosphate of the glycerate molecule. The seventh reaction is called ATP formation. This is a very important in the glycolysis process. In the glycolysis, very important position. This phosphate group in the one position is a transfer in the seventh reaction from the carboxylic acid carboxy group to ADP to give the 3-phospho D-glycerate. This is the very important reaction. Now, again the 3-phospho D-glycerate molecule to 2-phosphoglycerate conversion. The either reaction is an isomerization of the 3-phospho D-glycerate to phosphodiglycerate molecule. This is the 8th reaction of the glycolysis process, and 9th reaction is also very important to enter to the other metabolism pathways, dehydration process reaction. The 2-phosphoglycerate to phosphoenol pyruvate molecule. The next metabolism phosphoenol pyruvate form as a dehydration reaction from the 3-phospho D-glycerate molecule. This is also very important steps in the glycolysis. The next 10th step and last step also the formation of pyruvate and ATP production. This is the glycolysis pathway from the D-glucose to 2 molecules of pyruvate is concluded by the 10th reaction, which is transfer phosphate group from the phosphoenol pyruvate to ADP, thereby the giving ATP and the pyruvate molecule. What is the fate of the pyruvate reaction in the glycolysis? The fate of the pyruvate, the end product of glycolysis depends on the availability of oxygen and the metabolic need of the cell. This glycolysis totally metabolism at cytosol regions only is occur in the every tissue. The anaerobic conditions of the glycolysis process, in the process presence of the oxygen, the pyruvate enter to the mitochondria region, where it is converted into the acetyl-CoA by the enzyme pyruvate dehydrogenase enzyme, and acetyl-CoA when they enter the citric acid cycle also known as the Krebs cycle or TCA cycle. TCA means tricarboxylic acid cycle, where it is the undergoes further oxidation of the produce the ATPs and NADH and FADH2 through a series of enzymatic reactions in the glycolysis and Krebs cycle and citric acid cycle. This process known as the aerobic respiration maximize the energy of yield from the glucose metabolism. The primary mode of the ATP production in the most of the aerobic organisms. From anaerobic conditions, the glycolysis pathway, how it is worked out in an anaerobic condition, the absence of the oxygen, the glycolysis pathway, how it is going to conversion, what are the molecules production. So, now absence of the oxygen molecules under condition of the limited oxygen availability under the intensive exercise excess on hydroxy tissues, especially the pyruvate is converted into the lactate through the process called as a lactate fermentation process in an anaerobic condition. This reaction is catalyzed by the enzyme lactate dehydrogenase enzyme in anaerobic condition of the species, which is reduced by the pyruvate to lactate will oxidize NADH to NAD plus molecule, that is lactate fermentation allow the glycolysis to continue by the replenishing the pool of the NADH plus molecule to convert to the NAD plus required for the oxidation of the glycerol 3 phosphate, and ensure the continued the procedure of the ATP production and ability to low efficiency compared to the anaerobic respiration process. But the fate of the glycolysis is important, you are observed in that aerobic condition and anaerobic condition of the organisms. What is the physiological significance of the glycolysis role in the different tissues? In our body having different tissues and different tissues having different cells, organelles, the cells made up of different cells. In the different cells, the glycolysis mechanism, how it is going. Now, we are looking to the muscle cells tissues. The muscle cells having the high energy demand, the particularly during the intensive physical activities such as exercise, glycolysis play a crucial role in the meeting these the energy demand by the rapidly generation ATP through the breakdown of the glucose to pyruvate molecule. During the period of the high energy demand, such as during the stress exercises and the glycolysis in the upregulated by providing the necessity of the ATP to fuel muscle conditions. These allow to the muscles to maintain their functions and performance even when the oxygen availability is limited, as the case of the anaerobic condition. This is the exercise of the muscle cells metabolism required for the ATPs and pyruvate, how it is going to break down the glucose to pyruvate and utilizing ATPs. Now, liver cell glycolysis, which is the central metabolic organism, playing the key role of maintaining the blood glucose levels and regulating the whole body energy metabolism. Glycolysis in the liver serve multiple functions, including the glycogen storage and glucose production, and through the gluconeogenesis process also is very important. During the fasting and period of the increase the energy demand, and glycolysis is upregulated in the liver to produce the glucose, which can be released into the bloodstream to supply the energy to the other tissues such as the brain and red blood cells. So, this is important of the livers. In the liver having a big organ, and it will go more glycolysis process, then produce the more ATPs and energy sources and supply to the different organs. So, this is the important in the glycolysis mechanism in the liver cells. Now, blood cells, what is the role of the glycolysis in the blood cells? The blood cells erythrocytes lack of mitochondria and rely of the exclusively on glycolysis from the ATP production. This make the glycolysis essential for the survival and function of the red blood cells, which require the constant supply of the ATP to maintaining the membrane of integrity and transport of the oxygen throughout the body, and the efficiency of the glycolysis in the blood cells, and circulate of the maintain of the cellular homeostasis and ensure the optimal oxygen delivery of the tissues. This is the important of the RBC cells. The RBC cells is always supplied to the oxygen from in every tissue and every cell. So, this is the important of the glycolysis in the RBC cells. In brain region of the cells, what is the importance of glycolysis in the brain region? Now, we are looking to the brain region. The brain is one of the most metabolitic activity organ in the body and rely heavily on the glucose as a primary energy source. The glycolysis is the brain provide the ATP required for the neuronal functions. The including the neurotransmitters and synthesis and synaptic transmission and iron transport, because of neurons having the limit glycogens storage and cannot oxidize the fatty acids, especially in the brain region. The oxidation of the fatty acids cannot oxidation for energy levels. The glucose is essential for the meeting of the high energy demand of the brain under the normal physiological conditions. This is the importance in how the brain cells is going to activated. The what is the important glycolysis metabolism of our glucose enter to the brain cells, and it will how it is transportation and of the neurotransmitter regions, how it is work out in the neuron system, glycolysis pathways. Now, glycolysis we are discussed about glucose to pyruvate conversion and different 10 steps of the reaction, and after we in different cells having what is the importance there, how it is the glycolysis after pyruvate and glucose ATP supplying. Now, the glycolysis process diseases, the mean abnormality of the glycolysis pathways metabolism substances. If the diabetes is the very popular nowadays, the glycolysis is going to main play diabetes. The diabetes mellitus is a metabolic disorder characterized the chronic hyperglycemia and the high blood sugars, the resulting of the defect of the insulin secretion. The insulin action are both, and dysregulation of the glycolysis is a common for the future of the diabetes and contributed to the pathophysiological of the diseases. This is the importance of the glycolysis pathway in the diabetes, and what is the disadvantages, what is the advantages, how do we maintain the glucose levels, how we maintain the food intaking process, what foods maintain, how we are going to eliminated of diseases of the diabetes. There is so many people going to suffering of the without idea of the glycolysis pathways. They are taking very foods, so many foods. So, the how much is required for the cell, the glucose and ATPs, we have to discuss about the diabetes 1. Now, autoimmune destruction of the pancreatic beta cells lead to the insulin deficiency, impairing of the glucose uptaking and metabolism, insulin sensitivity tissues such as skeletal muscles and adipose tissues. As a result of the glycolysis dysregulation leading of the reducing glucose utilization and increase the glucose and production by the liver through the gluconeogenesis process. This is the understanding of the diabetes 1 condition, the glycolysis pathway, how the main role playing. We have to go analysis of the skeletal muscles and adipose tissues regions and brain cells and RBC cells regions and supplying the content through the glycolysis process, glyconeogenesis process. This contribution to the hyperglycemia and the development of the diabetic complications such as neuropathy, retinopathy. So, based on that we are medication, we control the glucose levels and glycolysis metabolism, glyconeogenesis process. In diabetes 2, what is the role of the glycolysis pathway? The insulin resistant and impairing of the insulin secretion lead to the glucose intolerance by the hyperglycemia. Insulin resistant in the peripheral tissues and disrupt the glucose uptaking and the metabolism lead to the compensively hyperglycemia and dysregulation of the glycolysis process. In this, liver and insulin resistant promote the gluconeogenesis and glucosis process, and further exacerbating and the hyperglycemia and dysregulation of the glycolysis and glucose metabolism contribution to the development of the insulin resistance. Certain variations of the percentage of the glycolysis, hyperglycemia, glycemia, dysregulation of the metabolic conditions, the diabetes 2 is a condition is there. This is the structurally understanding the glucose 2 pyruvate molecule, how the enter to the glucose and glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 bisphosphate, and aldose presents in the glycerol 3 phosphate, and it will convert to 1 3 bisphosphate glycerate and phosphoenol pyruvate and pyruvate molecule. In these pyruvate molecules, they are going to enter the different regions, and this is the diabetes, how diabetes playing the conditions, and tumor suppression regions also, how the target the molecule of the glycolysis in the tumor suppression of protein P53 and oncogene production CMYC and transcriptional factors HIF1 alpha also involve the and inhibited the of the content of the glycolysis. Now, coming to the overview of the citric acid cycle. Now, previous we are discussed glycolysis process. Now, citric acid we are going to discuss. How importance in the citric acid cycle is important has not be one, not two, but three different names in the common usage today. The name will be primarily used here. The critical citric acid cycle refers to the first molecule the formation of the during the cycle of reaction, the citrate or it promoted from the citric acid. However, you may also hear this series of the reaction called as a tricarboxylic acid TCA cycle. For these three carboxylic groups on the first two intermediate or the Krebs cycle after the discover the Hans Krebs. So, this is the name it will come by different names is there, TCA cycle, Krebs cycle and citric acid cycle names is there different. Now, coming to the citric acid cycle is the final common pathway for the oxidation of the carbohydrates, fatty acids and amino acids. In this cycle, the citric acid is the used for the generate the energy through the oxidation of the acetyl components of the acetyl CoA derived from the carbohydrates, fats and the amino acids. So, these are our biomolecules, then they are going to enter the breakdown and enter to the our citric acid cycles. So, overview of the citric acid cycle is the eukaryotic and citric acid cycle is taken place in the matrix of the mitochondria, especially citric acid cycle is at location at mitochondria region of the matrix. Just like the conversion of the pyruvate to acetyl CoA in a prokaryote, these steps both taken place the cytoplasm region only. The citric acid cycle is a closed loop, a last part of the pathway reform the molecule used in the first step. The cycle including the 8 major steps is there in the citric acid cycle, that is we are going to discuss 8 steps. This is the our mitochondria cell in the cell region, in the cell this is the one organelle of the mitochondria. In this the glycolysis process after linking the citric acid cycle, Krebs cycle and after oxidation and phosphorylation process, then the Krebs cycle it is produced 2 ATPs and oxidative phosphorylation, then 30 to 36 ATPs is produced. In the glycolysis only we discussed in the 2 ATPs are produced in the glycolysis pathway. Now, this is the exactly the pyruvate molecule enter to the mitochondria membrane, the pyruvate is converted to the acetyl CoA, then acetyl CoA is entered to the citric acid cycle. Now, this is the metabolism, then citric acid 2 again it will enter to the different pathways. This is the exactly the acetyl CoA molecule is entered to there, the C2 carbon containing molecule, the acetyl CoA and citrate molecule attached to there, then it will converted to in the presence of citrate synthase enzyme, it will synthase enzyme, it will convert to citrate and citrate to isocitrate molecule conversion in the presence of enzyme, and the isocitrate molecule is going to alpha ketoglutarate molecule. The alpha ketoglutarate molecule is again it will convert to succinyl-CoA enzyme, CoA molecule. The succinyl-CoA synthase enzyme in the presence of GTP to conversion, GDP to GTP conversion, then CoA is going to removing, then acetyl-CoA is converted to succinate molecule. The succinate is converted to at fumarate molecule. The fumarate is again to malate molecule conversion in the presence of fumarase enzyme, and the malate is converted to the malate dehydrogenase enzyme. It will convert to oxaloacetate molecule. The totally in this the oxaloacetate molecule again it will come 4 carbon containing molecule. Now, the oxaloacetate molecule is again it will break down and converted to the citrate molecule. The citrate is the 5 carbon containing molecule. Now, the total in the reaction, the total reaction mainly 8 steps is there. The citrate, isocitrate and alpha ketoglutarate molecule and succinyl-CoA and succinate, succinate to fumarate, fumarate to malate, malate to oxaloacetate molecule. These are the product, but the first molecule is called as a acetyl-CoA. The acetyl-CoA is coming from pyruvate glycolysis and enter to the mitochondria region in the membrane region, then membrane to enter to the mitochondria, then it will synthesis of the citric acid cycle or Krebs cycle or TCA cycle. Now, this is the overview for understanding basics. For understanding basics, the acetyl-CoA is a 2 carbonic molecule and CoA is there, the removal of CoA, the 6 carbon of the molecule of the citrate and citrate to it will convert to the different steps is there. Now, in the production of the ATPs, any importance in the citrate molecule, the total 10 ATPs per cycle is going to produce in the citric acid cycle. This is the acetyl-CoA and citrate, citrate to isocitrate, isocitrate to succinyl-CoA, succinyl-CoA succinate, succinyl to succinate, succinate to fumarate. There is different pathways is there for understanding in different levels of the degree level, intermediate level and PG level. There is pathways is going to different structures is going to showing. Now, we are going to discuss steps in the citric acid cycle. First step of the citric acid, the acetyl-CoA join with the 4 carbon molecule oxalate and release of the group as a forming of the 6 carbon molecule called as citrate. The second step is called as a second step, the citrate is converted into the isomer and isocitrate. In this circulate the 2 steps crossing inward the first step, the removal and then the addition of the water molecule, which is way to the citric acid cycle, sometimes describe the having the 9 steps rather than the 8 listing here. So, this is the steps for second, and third step in the isocitrate is oxidized and release the molecule from molecule of the carbon dioxide and leaving the behind the 5 carbon molecule and alpha ketoglutarate, and during this step ATP plus reducing the form of the NADH. The enzyme is called as catalyzed this step isocitrate dehydrogenase enzyme is an important enzyme, is important in the regulating the speed of the citric acid cycle. This is a very important molecule enzyme, the isocitrate dehydrogenase enzyme. Now, the fourth step is the third step in the case of the alpha ketoglutarate, that is oxidized reducing NAD plus and Na to NADH molecule, releasing the molecule from the carbon dioxide in the process. The remaining the 4 carbon molecule pick up the coenzyme A, the forming of the unstable compound succinyl-CoA. The enzyme catalyzes the steps. The alpha ketoglutarate dehydrogenase is called as an important regulator of the citric cycle. The fifth step is the CoA enzyme removing succinyl-CoA is replaced by the phosphate group, which is then transferred to ATP to make the ATP. In some cells ADP to ATP is conversion, ADP to ADP, and used for the instant of the ADP to form of the GTP also, guanosine triphosphate. G mean guanosine, our nucleic acid. Now, the production, the 4th carbon of the molecule producing the step of the called as a succinate, succinyl-CoA to succinate is coming. The ATP is similar to the ATP, both serve as the energy source, and the two can be related to interconversion, which is two molecules produced during the citric acid cycle depends on the organism of cell types, and ATP is the made up of a human heart cells, but a GTP is made up of a liver cells. In different cells, different steps in the citric acid cells there. In the step 6, the 6 succinate oxidase formation another 4 carbon molecule called the fumarate. In this reaction, 2 hydrogen atoms with their electrons transfer the FAD and produce the FADH. The enzyme that carry out the steps embedded in the inner membrane of the mitochondria. The FADH can be transferred into electron directly into the electron transporter chains, and in the 7th step, water is added to the 4 carbon molecule of fumarate. The conversion into the another 4 carbon molecule called the malate, fumarate to malate. The 8th step, the last step, the citric acid in the oxaloate state, the starting 4 carbon containing generated in the oxidation of the malate, another molecule is NAD plus reduced to NADH in the process. Product of the citric acid cycles. These are products of the citric acid. Let us take steps, some of the accounting, and molecules is very NADH molecule, FADH2 molecules and ATP molecules, and single turn of the cycles, acetyl-CoA form molecules and NADH molecule, FADH2 molecule, ATPs and GTPs, and fumarates molecule and acetyl-CoAs molecules. Then where all the ATPs, you may be think that the ATPs output of the citric acid cell seem pretty unimpressively. All that work for just ATP or GTP. It is true that the citric acid cycle does not produce much ATPs directly. However, this is the in different organelles having different energy sources producing GTP or ATP or other form of the content. It can make a lot of the ATPs indirectly by way of the NADH and FADH generated. These electron carrier will connected with the last portion of the cellular respiration system, depositing their electron into the electron transport chain to drive the synthesis of the ATP molecules through the oxidase to phosphorylation. This is the overview of the citric acid cycle. Now, this is today we are going to discuss about briefly about the glycolysis and citric acid cycle.